If we look at Varroa mites right now, you can see it has a near worldwide distribution. This map was made in 2010. I will say that since then, we know that Madagascar and most of Africa, surprisingly, has Varroa mite, but they don't seem to cause as much trouble in Africa as in other places. Hawaii now has Varroa mite, of course. There's only a couple of small places that don't, like there's the island of Newfoundland that doesn't have some mites. We see a couple of islands still in the Caribbean that don't have mites, but mostly mites are everywhere we keep bees except for Australia. And so if we look at that, what you have to remember is that Varroa destructor, this mite that we're talking about, the Varroa mite, did not come from Apis mellifera, the European honeybee, which originated in this area. It originated with Apis cerana, which is the Asian honeybee. And in Apis cerana, Varroa caused very little damage. And in fact, not all Varroa mite in Apis cerana are the same. There are these different haplotypes, or these different varieties of Varroa mite. One of those is the Japanese haplotype, and that was the mite that actually was introduced into Paraguay and was the first South American and North American variant. And that variant is not very lethal. It's a very gentle mite. Unfortunately, there's also the Korean haplotype, and that Korean haplotype is what predominates the world today. It's a much more aggressive, it's a much more reproductive, and successful, if that's what you want to consider successful, type of Varroa mite than the Japanese haplotype. So it was first described. Now, if you read the literature, if you go back a ways, you'll find that we talked about Varroa as Varroa jacobsoni first, and that was because it was first described as Varroa jacobsoni on Apis cerana in 1904. And so when scientists name something, they have a specimen, the voucher specimen, and that gives its name. Well, it turns out that there are several types or species of Varroa, and the Varroa we had originally was not actually Varroa jacobsoni, and that's why it got the new name Varroa destructor. And that was because they found out it was a different variation. But both Varroa jacobsoni and cerana and destructor live well on Apis cerana. And Apis cerana is the Asian honeybee. It's a much smaller bee. It has much smaller colonies. In terms of honey production, it produces maybe 2 or 3 kilograms of honey a year, so it's not very productive. But the bees live with mites very handily. And one of the reasons they do is, as we'll review, or you'll learn shortly, is that the capping, the length of development time for these bees only means that mites that go into drone brood will be reproductive. Otherwise, the workers develop so fast that even though if a Varroa mite invades that cell, it won't be able to have any children. Also, if that drone brood has more than one invading mite, somehow the bees sense that, and they'll cap that drone brood very heavily with a heavy capping, and that prevents that drone from emerging. And you see these distinctive little holes there. And so basically, they're entombing that drone and the mites in the cell, and then both the drones and the Varroa die. And so this Apis cerana has developed these behaviors which allow it to control the mite in the natural habitat. However, we manage Apis mellifera, the European honeybee. And so that mite, how did the mites then, what's the story of the mites getting onto and spreading? And I think that the point of this is to show that we have to be really careful about bringing stuff in, that people have done more to spread this mite than anything else. And so what happened was, we started here in Indonesia in 1900s. There was mites up in here with Apis cerana, but then the Germans, or Russian soldiers, brought Apis mellifera, the European honeybee, into Asia, and the mite jumped species. And then the Russian soldiers brought some of these colonies back into Germany, and we can actually base the spread of mites distributing throughout Europe because they were right beside a military base outside of Berlin, and that's where the mites came over. So the Russians brought them over to Germany, and then it spread throughout Europe, And then we've had introductions into Brazil, and then in the 80s we saw the introduction into the US and Canada, Hawaii 2007, so in the near past, and New Zealand in 2000. And so we can see that it spread sort of internationally because of human trade. So what causes it? Well, we can see the shipment of queens in packages. We've already talked about that a little bit. We also know that moving colonies is a great way of moving things around the world. And of course, we are reliant in this country and many other places on a movable pollination force. And so, as Randy explained, I think, well in his talk, half the colonies in the country are in almonds. And that means that if there's any new disease or any new problem or parasite that gets introduced in the country, and they go into the almond orchards, they're across the country in that next year. And so we have to be on the lookout for new diseases very vigilantly in order to stop it before it becomes a national issue. Now, one of the things that always amazes me, we talk about trucking our bees over long periods of time, but I think that to give it to some perspective is us moving bees from Florida to California is the same as moving bees from South Korea to Bangladesh twice a year, or going from Brussels to Moscow twice a year. And so this is a huge amount of distance we travel with our bees several times in the year. And so a lot of people say, well, isn't it the moving that kills the bees? However, in our survey, we consistently show that people who migratory beekeepers lose fewer colonies than non-migratory beekeepers. And so certainly there's probably some stress involved in bees, especially if you bake them or cook them or don't water them when there's a need. But those factors are manageable. We're able to outmanage some of those stresses, and that's an important factor. And so certainly we think that some of the spread, especially in Europe, can be traced along the beekeeper migratory rates. In Europe, there's a big migration into the forest to collect honeydew, the black forest for honeydew honey, and you can see that. And certainly throughout the US, we saw this spread. No doubt it was hastened by the migratory nature, but at the same time, that migratory nature is what makes the industry necessary. But even without people, mites are able to spread pretty quickly. So this was a great study done in Germany where they had an infested colony, and they watched colonies placed at several time points and distances away from that hive to see when they would become infested. So within 1 month, you saw colonies within 100 meters infested. Within 3 months, is that 3 months? 2.5 months, 500 meters or 1,600 feet away were infested. And in 3 months, 6 or 7 kilometers away or 4 miles away were infested. So this is a huge ability to move very quickly across the landscape. And that's because a lot of those mites that are moving are on drones. And drones are famously unfaithful. They will go to any colony that they want. And so they'll fly out and they'll go to other colonies. And so a lot of this is driven probably by robbing and probably by drifting, but a lot of it is drone drifting. So let's now delve into that sort of the biology of the bee or the Varroa mite and how it feeds. So what we have is we have the mites, and they live on the adult bees. That's the phoretic stage. They're on there, and then what they do is they when the bees are feeding the larva, they sort of come in and take a smell. And if the smell is right, they'll jump off and they'll bury underneath that larva and live in that brood food until it gets capped. And then that mite crawls up and she starts to lay eggs. We'll talk about all these steps in a little bit more detail. And eventually, over here, all the female mites that she's laid, all her daughter mites that are fully mature, so they're fully sclerotized, their outer cover is completely solid and brown, and that's because otherwise it gets dehydrated. Remember, that exoskeleton is to keep it wet from drying out. All the mature female mites will come out with that bee. So let's look at all these stages and see if we can learn a little bit of something with that. So we're going to go with this video.
We'd like to introduce you to the Varroa mite.
Oh.
Well, to be precise,
That's really loud. 1 second. Sorry. I'll I'll calm that down. What the heck? This video is courtesy of Nod, who I think are here. This is a great little video. So this video is going to introduce some of the bigger themes that we'll be talking about throughout this.
We're waiting. Are there any questions that I won't be able to answer?
Right here.
Question.
For Dennis, now that he's not being informed, but is asking about the bee losses in the summer, is there any way to go back to get an idea of how long that's been going on at that rate?
And why?
So, well, I mean, that's a really good question. Can we go back and figure out what was, like, has it happened before in the summer? And that's a real big problem even in winter losses before we did the survey, because it's all anecdotal. Like, there's no consistent survey. And I think that's why it's great that NAS is now taking over the survey. I will emphasize, NAS gets $2.5 million a year to do the survey, and we got between 30 and 50,000. So hopefully that will be a very robust survey, and it'll be interesting to see how the results compare. But certainly we've worked closely with them, and they're continuing to do the summer loss. In terms of can we go back and look at, well, what are the drivers? Our management survey was set up to look at winter loss, which I'll show you here, but there is a way, we just have to do it. We have to connect management surveys from year to year, which is a little bit more complicated, but we think we can look for some of that. We just have to finish the... Oh, sorry. The question was, can we go back in time and figure out what was the normal summer loss sort of historically? And then also, how do we figure out what the drivers are for those losses in the summer?
We'd like to introduce you to the Varroa mite. Well, to be precise, we'd like to introduce you to millions of Varroa mites. Why so many? Well, when it comes to the Varroa mite, there's no such thing as just one. And if you'd like to see how an infestation starts, you need to look no further than your own bees, especially drones, as they're pretty social and like to travel between hives. Since Varroa mites don't have any wings of their own, they slip into hives by hitching a ride on the backs of adult bees. And for the lucky mite, the trip includes an in-flight meal, as Varroa mites will begin feeding on bee blood within a few minutes of clinging to the bee. Once they've entered the hive, Varroa mites slip undetected into the vulnerable, uncapped brood cells. This is where the mites lay in wait until the bees cap the brood. Once a cell is capped, the mother mite, like a tiny vampire, climbs atop the cocoon of the developing bee, tears open a hole, and begins to feed on its blood. Within 3 days, the mother mite lays her first egg, which always develops into a male. Then, she lays one female egg every 30 hours over the next week or so in her newly acquired home under the brood cap. As each of these female mites mature, they mate with their brother. By the time the baby bee develops and leaves its infested cell, as many as 3 fertilized mites will emerge with it, and the cycle continues. Using this strategy, the Varroa mite population can grow as fast as the bee population it feeds on. But when summer ends and the bee population declines, the hive is left with a huge mite population, and that's dangerous. Too many mites in a hive will overwhelm and kill entire bee colonies. So what does that mean for everyone's favorite insect, the honeybee? Well, honeybee colonies with heavy mite infestations can't effectively pollinate or produce honey because they suffer from diseases and viruses transmitted by the mites. In fact, honeybees suffer from as many as 20 different mite-induced viruses, including the devastating deformed wing virus.
So I bring that video up because what we're going to do now is sort of take, that was a really good summary video, and just go up to, I think it's in the Nod Apiary or website. You can watch that video yourself. But I want to now tear what we've just seen apart a little bit and look at it really carefully, because I think it's really important, as I said before, for us to understand some of this biology. So what we heard, so here we have the life stage of a worker bee, right? So it's 3 days as an egg, then we're at this larval uncapped stage. This is the growth stage where the nurse bees are feeding them like crazy. This is when they're going to get fat. It's the only stage at which the bee grows. Then it gets capped and becomes the pupal stage. So this is when it spins a cocoon, that larva goes to the bathroom for the first time. And then it undergoes this really quite amazing transformation, because basically all of its organs dissolve and becomes this big bag of fat. And that fat then gets organized into legs and arms and hearts and organs and all of that stuff. So this is a really quite a remarkable time for the pupal bee. And then the adult emerges somewhere on day 21. Well, here what we see is that the female mites on a nurse bee enters that cell a day or two before capping. And then after capping, 3 days later, or 60 hours later, it lays its first egg. And then it lays an egg every 30 hours thereafter. However, so these are the different stages of the mite. Only those female mites that get sclerotized over here are going to survive hatching out with the emerged worker bee. This is a courtesy of Peter Rosenkrantz. He's really I think one of the world experts on Varroa from Germany. He's done a lot of work on this, and he's sort of just done exactly the same thing we've just talked about, but on a different scale. So you can see that first egg is a male egg. Now the reason it's a male egg is that first egg she lays is unfertilized. And so it's exactly like worker or queens, right, or honeybees, where fertilized eggs become female and unfertilized eggs become male. And so that male comes out and it becomes this sort of white male, and it will always remain white. They die after the adult bee emerges. But then all these female mites' eggs get laid and they start to mature. And they will then mate with their brother as a result of that. So one mother mite will result in 1 to 3 daughter mites from each infection. I will say, though, that some of these mites don't get mated, and so they're virgins. Now that means that they can go in and that they can invade another cell and they'll lay male eggs and they can mate with their son. But generally those are not very successful. So about 10% of mites haven't been mated and will just lay duds, basically. They'll just have males and they won't be able to reproduce. And so there's been quite a bit of work on trying to figure out, well, are there ways we can induce this sterility so that the male or the mites aren't mating? And so if this is another way of looking at it, and this is really quite asking, so we have this phoretic phase. Phoretic phase means basically hitchhiking phase. So we have these mites, they're phoretic, and as that nurse bee goes around and feeds larval bees, those mites go out and they actually smell the cap, they smell the larva. And if the larva smells right, they'll stay in that cell. If it doesn't smell right, they leave with the worker or the next worker and go on to another cell that's the proper aged cell. So they're smelling that larva and knowing the right age. And so right after it gets capped, that 5th instar larva releases a special scent. And that scent allows the Varroa's ovaries to start activating, and it starts to produce its first egg. If it doesn't get that shot of smell in that first 24 hours, the ovaries don't develop and she'll never start laying eggs. And so that's a really important step there, is that activation. And then we see, of course, then it becomes, as it matures, it becomes a proper egg. And you can actually see here that in that immature female, you can see it's still white, how big those ovaries are compared to the rest of the body. And then it'll have a whole bunch of offspring, and then the males will mate with the females. This is how they know about this Varroa. This is a really cool experiment where they took the scent of a 5th instar larva or a prepupa, and they put Varroa mites here, and then they watched what happened. And so what happened is basically, unless they were offered that smell of a 5th instar larva, you didn't see the ovaries develop. So this is a really great study that was done out in Germany. This is really going slow. So mating actually is also an interesting thing. Well, yeah, some things go slow. But so you can see the male is much smaller than the female. You can see the female over here. He actually courts her and will tap her and do a whole bunch of, hey, I'm a nice guy sort of thing. And then he actually spends a lot of time trying to find out where their sex organs meet, and eventually does that. And they actually, because this is her sex organ here and that's his, and so then they have the transfer of sperm, and then she stores all that sperm, just like a queen bee does, to fertilize the eggs for the rest of her life.
Does she have to be an adult before that happens?
Well, she mates still while she's not completely sclerotized. So it's just before she gets sclerotized. She can't mate when she's sclerotized as well. I think they can still mate because she can mate with her son sometimes, but the best time is just before they become the final molt. So we've seen this slide already. So basically, this explains, if we think about that, the fact that it's the length of time under the cappings is what limits the number of mature mites that can develop, right? Because the longer you're under the capping, the more your daughters are going to be fully sclerotized. And so this explains why Varroa prefer drone brood. Because if you think about it, if your whole purpose in life is to have as many kids as possible, then if you can get under a drone cell that's capped for 13 to 16 days, you will on average have 2.2 to 2.6 kids that are fully sclerotized and able to survive with the drone that emerges. In workers, they're capped only between 12 and 12 and a half days, which means they have half as many kids that successfully emerge. So that's why drones are this great breeding ground, and that's why Varroa mite really do prefer drones. Varroa mites do invade queen cells, but queen cells are not capped long enough to have any viable offspring. And so even though you might see it there, they're not reproducing successfully in those queen cells. So I think this is the 7th inning stretch. So we're gonna take a short break. We have to all sing along though for the 7th inning stretch. Do you guys know this guy? So we're going to take a 20-minute break now, and then we'll come back for another 50 minutes, and we'll take another 20 minutes.